Please join me in welcoming to the Distinctive Voices podium, Dr. Lauren O'Connell.
Hi, everyone. Thank you for coming. And thank you for that really wonderful introduction. So I'm excited to share with you tonight our research on the evolution of pair bonding and parental care across many different taxa. And I'm hoping to convince you that we can learn a lot from cold-blooded animals and that you have more in common with a frog than you thought before coming here tonight. And so my lab studies social bonds, and in particular social bonds within family units. And I became really interested in this question when I started my own family unit, shown here. And if you think about this family unit or your own family unit, there's a lot of interesting social interactions happening here. So for example, there's pair bonding, or the ability to tolerate someone else in close proximity for long periods of time. This was very well tested in the pandemic, as many of you know. And then there's also parent-offspring interactions. How do you choose, or how do parents choose how much to invest in current versus future offspring? And from the offspring perspective, how do you solicit care from your parents? And then anyone with kids or siblings will know that there's a rich diversity of behaviors among siblings, play, resource competition, and sometimes aggression. And so we study all of these different interactions within family units. And understanding these interactions both within families and outside of families is really important for human health. So from a mental perspective, people with strong social bonds are happier, they have reduced stress, and it also impacts us physically. And so there was a meta-analysis done on humans that showed that social relationships and the strength of social bonds is actually one of the top predictors of longevity in humans. And so, in my opinion, it's really important to understand how social bonds are formed, how they're maintained, and how it influences our mental and physical well-being. And so most of what we know about social behavior and social bonds actually comes from laboratory rodents. And this animal has given us really an unprecedented view and depth into the neural basis of these behaviors, thanks to a lot of tools that people have developed for studying them. And so, for example, we know a lot about maternal care, how maternal care is implemented both from a physiology and neural perspective. And this is because, decades of scientists keeping mice in laboratories has selected for high quality maternal care. Moms that eat their babies don't make it, they don't contribute to the next generation. And so a lot of this work has shown that this brain region called the hypothalamus is really important in maternal care and mother-offspring bonding. And we'll come back to the hypothalamus when we talk about frogs in a little bit. But this focus on using laboratory rodents has, it's given us incredible depth, but it's also limited the number or the different kinds of social bonds that we can study. So for example, we don't know a lot about the neural basis of parental care in males. Male laboratory rodents don't normally show paternal behavior. We don't understand a lot about social bonds between, in the context of pair bonds. And we don't understand a lot about how bonding happens from an infant perspective. And so these are the gaps that my research group is trying to fill. And so there are two questions we're going to tackle tonight. One is, are the neural pathways promoting parental care shared or distinct in males and females? Is it the same mechanisms happening in the brain between the two? And then the second question we'll look at is, are the mechanisms that promote parental care the same as we see in pair bonding? And so there is a beautiful diversity of animals that live in family units. And so family units are phylogenetically very widespread. if we start from the right here in these different columns, we have mammals, reptiles, birds, amphibians, fish, and then invertebrates here on the very left. And they all display different kinds of parental behavior, which I have shown for you. At the top is male uniparental care, in the middle is biparental care, and at the bottom is female uniparental care. And I want to note a couple things. So one is that there are no uniparental male mammals. The definition of being a mammal is lactation. And so moms, they kind of have to be involved. But that means that there aren't any uniparental male species in the mammalian clade. A lot of work, though, is done on paternal care in rodents, and this is usually done in Microtus voles, which we'll talk a lot about. But in this context, when male mammals do show parental care, it's usually in the context of biparental pair bonding species. And so because parental care and pair bonding are happening at the same time, those are really difficult to dissect, to tease apart in the brain. And so what I decided to do was to study a different clade of animals that showed a lot of diversity in these different kinds of behaviors that had male uniparental care, biparental care, and female uniparental care, so I could tease apart what promotes parental care and what promotes pair bonding in the brain, which is not possible or very difficult to do in a mammal. And so what I found in the literature were these really wonderful amphibians. These are dendrobated poison frogs. And what I really liked about them was that, well, besides from their color and being quite beautiful, was that they show this incredibly rich diversity in parental care strategies, all within closely related species. That, and then they live in very similar environments, so they have similar ecologies. And so I thought they were a great system in which to explore this question. And so each species shows different flavors of parental care, and what I'm showing you up here is different species where the sex of who's caring for the offspring differs. And so if you look all the way over here on the left in Alabates femoralis, what you'll see is that this male is carrying tadpoles on its back. So this is the school bus method of tadpole transport. He's transporting a lot of tadpoles at once, whereas these other species are only transporting one at a time. And the reason for this difference is because they utilize different pools in their environment. And so here for Alabates femoralis, they take care of lots of offspring at once. They drop them in a really big pool of water where there's lots of food, lots of predators, but everybody gets along. They don't eat one another, and the tadpoles are pretty gregarious. But then as we start going to other species and they decrease the pool size in which the parents are using to drop their tadpoles off, we see a lot of other behaviors starting to emerge in these tadpoles. So one is that the tadpoles become cannibals, and so they will aggressively defend their tiny pool of water from other tadpoles, and then they'll end up eating one another. And then because of that, this is why you see these parents only transporting one. If you drop two tadpoles in the same pool, there will only be one later. So one it is. And then as you decrease the size of these pools even more, then these pools have very low resources, not a lot of predators, but there's not a lot of food, and so the parents have to come back and feed them. And they've evolved this really intricate communication behavior. And so before coming here tonight, you probably didn't think of frogs as really engaging, dedicated parents. And so I want to take you through one kind of bout of reproduction so you can get a sense of how these behaviors work. And so the example I'm going to show you is this frog, the mimetic poison frog, Rhinomatea variabilis. They're one of the few genetically monogamous species on the planet. They pair bond, and they work as a team to raise their offspring. They lay their eggs in the leaf litter, and the dad takes care of the eggs, keeps them moist, and when the tadpoles hatch, they are still aquatic, and so they have to make it to water, and their parents give them a piggyback ride to do so. And in this particular species, they place each tadpole in its own nursery. And its nursery are these little pools of water contained within plants in the rainforest. Because remember, the tadpoles are cannibals here, so it's important to place them individually within these pools. And because these pools are really tiny, they have evolved this mechanism to come back and feed the tadpoles. And so what happens here is that Dad will visit all of his tadpoles. He does this nearly every day. The tadpoles, to communicate that they need food, do this little dance display where they vibrate back and forth near the parent to communicate that they need food. They don't do this when they're not hungry, so it suggests it's an honest indicator of need. Dad jumps out, and then he calls for his mate, who's somewhere nearby. Mom comes, and if the tadpole dances sufficiently, she decides to feed the tadpole and these trophic unfertilized eggs. And so this happens once a week for several months, and they have multiple tadpoles going on here in the forest. And so I hope you can appreciate here just how much energy they're spending on each of these tadpoles and how much time it takes to rear some of these tadpoles in the wild. And so what we decided to do was to take this comparative approach and ask if there's a general parental care circuit in the brain, or is it different between males and females? And so we have a male uniparental species here, a female uniparental species, and a biparental monogamous one that I just told you about. And so to do this study, we have some frogs in the lab. And so they're arranged in tanks in pairs. And so we can, so this is the female here, and this is the male. And some of these species where males care for offspring, the females are much larger and much more aggressive. And so they have these shelters here and their tanks in a pool of water to place their tadpoles. And what's nice about doing this in captivity is that we can follow the behavior of both the male and the female. We also did a series of studies in the field, and so we have these long-term field sites in Ecuador and that are shown here, these enclosures here. These enclosures have several frogs per kind of square, and so we know who mom is, but we don't know who dad is, and so we were only able to collect females in this particular case. And so what we wanted to do to get a handle on the brain and how it promotes parental behavior is we needed to look for neurons that are more active. And so to do this, we use this assay where we're looking at phosphorylated ribosomes. And so the idea here is when a neuron is more active, it needs to make more protein, and it phosphorylates its ribosomes to do that. And this we can detect with an antibody and label the neurons brown. And so essentially what we're doing is we're labeling neurons that are more active brown, and then counting those neurons and comparing the number of brown neurons in our animals that are doing tadpole transport to the animals that are not. These are our control group. And then we're counting the number of active neurons in a bunch of different brain regions in the amphibian brain. And just a side note here about how we kind of determined what brain regions to count in is that in the brain regions that regulate social behavior in mammals are really well understood. So this includes brain regions that use dopamine and brain regions that use a number of other markers such as sex steroid hormones and neuropeptides. So many of you have probably heard of oxytocin and vasopressin, for example, and these brain regions have many of these different neuropeptide and signaling molecules. And because there is a kind of a map of where these are in the mammal brain, what I spent some time doing as a graduate student was mapping where all of these are located across vertebrate taxa. And so what I wanted you to take away from this is that the brain regions that regulate behavior are very old, and that they're similar in mammals, and we can find the same brain regions in amphibians. And these are the brain regions that we decided to focus on. And so what I'm going to show you here is our data for one species, and this is Dendrobates tinctorius, a male uniparental care species. I'm going to show the sex of the animal doing parental care here on the side. And I'm going to highlight the brain regions that are more active when the male is doing parental care. And so we could, and there's quite a lot going on here. There's a lot of different brain regions that are active, and we could have stopped there saying that, oh, maybe these are brain regions that promote paternal care. But the whole point of even starting this system was to do a comparison, a comparative approach. And so I want to highlight a couple things to you here, which is one, there are some brain regions that are only active in dads, only active in males that are doing parental care. There are others that are only active in biparental species. So maybe something about tag teaming parental care involves this brain region. But what we're going to focus on today is the similarities across all of these species. And so I want to show you, this was kind of cartoonized for you, and so I want to show you the actual data here, and starting off in the hypothalamus. So the hypothalamus is a brain region that regulates social behavior, and I told you in the very beginning that this brain region controls maternal behavior in rodents. And so this was really exciting to us that we're already starting here with a pretty old brain region that does a similar function in mammals. And so what we're looking at here is the number of active neurons, and I'm going to show you here, these are our control groups. So these are pairs that are not caring for offspring, and then this is a pair where the male is transporting. So I'm going to highlight the transporting individual here in orange. And what you can see is that there's a lot more active neurons in the animal that's doing the transport behavior compared to these other three. And then this data looks very similar in our other species where the animals that are doing this transport task have more neurons that are activated in the hypothalamus. And one more interesting thing I want to point out, and this is a common theme that we see, is that in the biparental monogamous species, we see that females mirror the neural activity of their partner, even though they're not actually doing that behavior. All right, the other brain region that we found to be correlated with parental care in these species was the hippocampus. And so the hippocampus in mammals is involved in spatial memory and navigation. And so we also found that the animals that were doing the transport task had more active neurons in the hippocampus. And so to us, this made sense because they actually have to remember where they place their tadpoles or where high-quality pools are located in the forest. And so one of the things that we do in our field studies is that we developed these frog pants. You can see here. So here are these different species. They're carrying tadpoles on their backs, and this is what the white arrow indicates. And then you can see that they have these little pants with these little transmitters attached, and it allows us to follow these tiny frogs throughout the rainforest to see, the secret life of frogs. And so it allows us to follow them around, and we have published quite a lot about their spatial navigation and behavior and how it links to parental care. And so far, we have some good hints about brain regions that might promote offspring transport, but we wanted to utilize or to pull out one more trick to utilize behavioral plasticity or behavioral flexibility in these species to test to see if similar patterns emerged. And so most of what I told you so far is we're studying the transporting sex and their non-transporting partner. But if you remove the mate, the animal that does the transport behavior, you can actually stimulate this behavior in the sex that doesn't normally do it. So for example, in this species, if you remove the male, the female will transport their tadpoles because the other choice is for the tadpole to die. And so even though we don't normally see this behavior in the wild, it's very rare, we can induce it to happen in the lab. And this one tells us something very important, that females in this species do have the capacity to do this behavior. They just, under normal circumstances won't take the time to do it. And so what we did then is we looked at neural activity in these animals as well. This is the data I've already shown you with transporting males having more active neurons. And then this is our sex reverse transport. So in dark purple is a female who's been induced to do the same behavior, and we see the same rise in neural activity here. For the hippocampus, we did the same thing. This is the data I've already shown you. And then in the reverse sex reverse transport, we see a similar trend, but it's actually not significant. And so what I've shown you so far is that we've used these comparative approaches across many species to show that there's overlap in neural mechanisms that promote tadpole transport behavior. And it suggests that the circuitry that is used for parental care is actually the same in males and females. The circuitry exists, but what's different is the stimulus that causes the circuitry to be activated. And so, for example, in our male uniparental care species, the female doesn't normally do this behavior, but they can be induced to do it under special circumstances. So all the machinery's there, but what activates it is kind of the next question. And so what we wanted to do next is to figure out what signaling molecules were important for coordinating care. And so to do something like this, we're going to use this same concept of phosphorylated ribosomes. And so ribosomes translate RNA into protein. And so they're holding on to mRNA as it's being translated into protein. And so we can use an antibody to isolate these ribosomes and their RNA attached to them. And then we can quantify the amount of RNA and what kind of genes they're encoding for. And so it allows us to create a molecular signature of parental neurons in the brain. And so we did this in a couple different species, and I'm going to show you one example here. And so this is data for the hippocampus. And what is going to be on the top are transcripts that are enriched in transporting males. So you can think of these as signatures of neurons that are active. And what's on the bottom are depleted in transporting males. So you can think of these as neurons that are kind of silenced. And so there are lots of genes being expressed in these neurons. And so we looked for signaling molecules that are expressed within these neurons. And so I've labeled a couple of them for you here. I'll point out a couple that I think are cool. So for example, here's the androgen receptor here, which may be important in male parental behavior. And then here's arginine vasopressin here, which I mentioned before as being a neuropeptide important in behavior. And so we did the same thing in the hypothalamus. We got a number of other genes that might be contributing to behavior. But what I really want you to focus on here is the only one that kind of overlapped between the two. And that is a peptide called galanin. So galanin is typically associated with feeding behavior. When I first saw this result, I was like, womp womp. Like I wasn't very excited about it because it has a lot of research into this peptide and like the obesity and metabolism field. But around this same time, a paper came out from Catherine Dulac at Harvard that showed it was also associated with paternal care. And so usually in the lab, male mice, if they're presented with pups, they will eat them. And so they'll show infanticidal behavior. But the Dulac lab showed that if you stimulate galanin neurons, you can turn a male from an infanticidal male into a caring dad with like the flick of a switch. So they'll switch from infanticidal into this licking and grooming behavior. And so we were really excited about this because we thought, well, perhaps, we also see this infanticidal behavior in frogs because, these males are caring dads, but if they take over the territory of another male, the first thing they do is eat all the eggs. And so we hypothesized that perhaps galanin was this switch from this infanticidal behavior into this offspring care behavior. And the other thing that was exciting to us about this is that the circuits are very similar in male and female rodents. And so we thought this might be some link to conserved parental care circuitry. And so what we did in our frogs is that we labeled neurons in the hypothalamus with different markers. And so I'm showing you in red are galanin neurons, and then in green are neurons that are more active during tadpole transport behavior. And what we're interested in are the ones that are yellow, the ones that are co-localized. It tells us what percentage of galanin neurons are more active. And when we did this, for example, we found that galanin neurons are more active in animals that are displaying parental behavior compared to animals that are not. And so to summarize what I've shown you so far, we found that galanin is associated with parental care in amphibians similar to mammals. And what I think is really amazing about this finding is that even though amphibians and mammals separately evolved parental behavior, they're using the same neural toolkit to do it. And so, which I thought, that blew my mind. So I thought that was really amazing. All right, so to summarize what I've shown you so far is that, we started off with this system asking the question, are there different circuits for parental care in males and females? And so what we have found is that the circuits are actually very similar, and what's different is what activates these parental toolkits in different contexts and different species. And this is the next question that my lab is trying to figure out. And so with the rest of my time with you, I want to focus on pair bonding and ask if the mechanisms that promote parental care are the same or different as in pair bonding behavior. And so we're coming back to this idea that it's really worthwhile to look at the behavioral diversity present in other taxa because it lets us tease apart behaviors that would otherwise be very difficult to study in traditional animal models. So for example, most neuroscience research in pair bonding comes from Microtus voles, which are shown here. They both pair bond and they show parental behavior. So it's difficult to disentangle the two when you're trying to figure out the neuroscience behind these behaviors. And so leaning on other taxa, we can study how and why different species pair bond without parental behavior as a confound. But first, let me summarize kind of the field of pair bonding behavior in voles before we come back to other cold-blooded animals. And so vole research has really highlighted three neural signatures of pair bonding. And so this field, and especially with Microtus voles, it's also very comparative, meaning that they compare species with pair bonding to those that do not. And so, for example, the prairie vole pair bond, they like to hang out with one another, they raise their offspring together, whereas meadow voles do not want to hang out. They do not cuddle in the laboratory like prairie voles do. And for example, once after they've mated, they actually prefer a new partner, whereas prairie voles, after they've mated, will prefer only their own partner. And so there's this very selective pair bond that happens in these animals. And so there's been a lot of research looking at the neural basis of pair bonding. And so one of the things that people who work on voles have found that there's this density of neuropeptide receptors that varies across species. And so the neuropeptides that they've really shown to play a role in this behavior are oxytocin and vasopressin. And so when you look at a slice of a prairie and a meadow vole brain, what this shows is the blacker this is more oxytocin receptors present in that brain region. And so you can very much see immediately the differences between the two species. There's a lot of oxytocin receptor here in prairie voles, but not meadow voles. And this is similar to what's happening with the vasopressin receptor. There are lots of species differences in receptor density. And there's a very similar result when we look at dopamine and dopamine receptors being required for pair bonding. And so what we wanted to do is that, well, people have noticed that there are similarities in the mechanisms between pair bonding and maternal care, mostly in this research involving rodents. And so some people have proposed that pair bonding actually evolved from the maternal-infant bonding because there's so much neural similarities between the two behaviors. But this is a somewhat rodent-centric or mammalian-centric view because in other taxa, maternal care is not an ancestral feature like it is in mammals. You know, all mammals display maternal behavior, and so maybe this is why when you evolve pair bonding that you use this same circuitry to evolve pair bonding. But that's not the case in other taxa. And so what I'm showing you here is kind of this tree of vertebrate animals and that are labeled here. And in the pink, it's whether or not pair bonding is present and the percentage of species that show pair bonding behavior. So for example, 90% of birds show pair bonding behavior, whereas only less than 1% of amphibians do this. There's quite a lot of variation throughout the clades. So for example, over half of New World monkeys do pair bonding behavior. And so in rodents, and what we've learned from Microtus foals is that these different factors are involved: oxytocin, vasopressin, and dopamine. And the reason for pair bonding is this biparental care that they show. It's advantageous and the offspring are more likely to survive if the dad is present, helping raise them. But what we're going to focus on today is these other critters. Specifically, we're going to walk up the vertebrate tree and with examples from Chaetodon butterfly fish, from some poison frogs, and then we're going to end on these sleepy lizards here. All right, so what I want to do is I want to take you to our field site in Australia off Lizard Island. And so this is the view that you see when snorkeling on Lizard Island, and you can see that there's a lot of different species here. There's a lot of social interactions happening. This reef is relatively still high quality, but there's still a lot of resource competition on the reef. And what I'd like to show you this next video of is I want you to pay attention to these fish in the middle that are white with a yellow tail here. And so these are butterfly fish, and these two butterfly fish are pair bonded. And so they are hanging out together. You can see that they kind of have parallel swimming behaviors. They're following each other in a very parallel way. And what's happened here is that these butterfly fish, here, I'll play that for you one more time, if I can find my mouse. Oh, it's going to do it anyways. Okay, so these butterfly fish are corallivorous, meaning that they specialize on eating corals. And so it's thought that the reason that they evolved pair bonding was to be able to defend their patch of corals as a food resource. And so this is a research scientist in my lab, Jessica Nowicki, a picture of her doing field work. So she's following around a fish on the reef and recording its behavior with some underwater paper and pen. And I just wanted to give y'all a sense of what field work was like. And so, yeah, we're following around these fish, snorkeling. And so what we're looking for is what percentage of time they're spending with their partners and if they're doing parallel swimming. And so what we found, I'm going to show you just one species first, because what we wanted to do was look at the percent of observations where a particular focal fish, or the fish we're paying attention to, spends its time paired or alone or solitary. And so in this particular species, most of the animals are paired. They're spending time with just one other individual. And this is common across 3 other species that we were studying. But this is not the case for all butterfly fish. So we have some species that hang out in pairs and other species that live solitary lives. And so what we wanted to do was to see what was, if they're paired, do they spend more time with their partner or a non-partner? Is this pair bond selective towards one individual? And so what we found is that we were looking at percent affiliation time between a partner and a non-partner. And so, for example, in a pair bonding fish, they spend over 75% of their time with their partner, with this one other fish. And so this is very similar to the other pair bonding fish, but not to the solitary species. So they tend to hang out alone, not with a member of their own species. And then the other thing I think that is really important to note here is that, these species do pair bonding, but they don't do parental care. They actually mass spawn into the water column. And so it's thought that they evolved, pair bonding for this resource defense. But when we remove reproduction and parental care from the equation, we actually see a lot of diversity in who is pairing with who. So we see mostly male-female bonds in these fish, but we also see a lot of female-female bonds and male-male bonds within many of these species once, parental care is removed from the equation. And so the other thing I'd like to point out that to kind of reinforce this idea that pair bonding evolved for resource defense is that fish with a partner have more energy reserves. And so what we can do is we can look at their livers to look at the energy reserves that they have. And when we're looking at enduring pairs, so these are fish that have paired with the same individual over multiple years, as well as new pairs, they have more kind of energy reserves than do individuals that are solitary. And this is the case for a species called Lunulatus, and is what we found a similar result in other species. So we think that, being able to pair up, being able to defend your coral resources gives you more energy reserves, and then also leads to a lot of variation in who your pair bonded partner is. All right, so to summarize, our work in butterfly fish showed that they're mostly monogamous due to this resource defense behavior. I'm not going to talk about our work in frogs, although I would love to, but they do bonding for biparental care similar to mammals. And so what I'd like to end up with, though, are these monogamous reptiles here. And so, but first I want to take you to our field site. So this is in a different coast of Australia, but near the outback. And so what you can see here is that the terrain is very sparse. There are these bushes around. The sun is you can feel kind of the sun coming through this video. It's very hot. And there isn't a lot around to eat. And so there's not a lot of resources here. And so this is the terrain and habitat of the species that we're studying. And so the species that we're studying, you see the same kind of parallel walking behavior, these two like little lizard sausages, and they're just walking around, and they're following one another. And this is actually the only pair bonding reptile in the world. And they live for 30 or somewhere between like 40 and 50 years. And they will, they pair bond for, going on 20 years. Like as long as this study has been happening, they will stay with their partner. So this is a close-up. What is really nice about this system as well is that they do not provide parental behavior. So they pair bond. They will find their, after the lizard version of hibernation, they'll emerge, they kind of hang out separately. After they emerge, they re-find their partner to mate, they hang out for a little while, and then they go back to sleep. And so they do not provide parental care, which is a great opportunity for us to study how pair bonding happens in the brain without parental behavior, similar to the butterfly fishes. And so in this species, though, monogamy is thought to evolve to reduce disease. And so what I'm showing you here is a picture of a sleepy lizard, and these are ticks, gigantic ticks that are sucking the blood out of this lizard. And so it's thought that monogamy evolved to reduce parasite load because monogamous lizards have reduced parasite loads compared to promiscuous lizards and solitary lizards. And actually, divorces can happen if one of the individuals has too many parasites. And so what we wanted to do was to study, all right, where in the brain might this pair bonding behavior be happening? And so we looked in all the brain regions that are involved in pair bonding in voles, and we only found 2 that were brain regions that were more active during pair bonding behavior in this species. And these are both brain regions that are involved in dopamine signaling, which we were really surprised at because we thought that given the huge literature on oxytocin signaling and pair bonding that it would be similar here. And so we also did a similar thing as we did in Galanin, where we labeled the oxytocin neurons red, and then we labeled active neurons in green. And we looked at the percent of active neurons in pair bonded and non-pair bonded individuals and didn't find a difference between the 2. And so we also found similar results in frogs and fishes that I don't have time to talk to you about. All right, so to summarize here, the reason for pair bonding in skinks is thought to be disease, so reducing disease load or parasite load. And what we found is that when you look outside of mammals, that it seems to be alternative mechanisms that might promote pair bonding. And this is what we're trying to figure out now, is what are the actual neurons and signaling molecules that promote pair bonding whenever parental care is kind of out of the picture. All right, so just to summarize this part here, we found that dopamine rather than oxytocin seems to be involved in pair bonding across taxa. And so what we're learning from these cold-blooded animals is that there's this beautiful diversity of successful family structures in the wild, and that they have shared and distinct neural pathways that are facilitating these behaviors. So for example, I told you stories about single dads and single moms and how the brain regions and cell types that are promoting these behaviors are very similar. And I also told you stories about how when you remove reproduction from the, or offspring care from the equation, we actually see quite a lot of diversity in the kinds of pair bonds that animals are making and who is pairing with who. And so one of the major things that we've seen is that parental care and pair bonding mechanisms are actually very discrete. And so we're looking forward to learning more about the neural basis and physiological basis of social bonding from these unusual animal species that have taught us so much about why behaviors evolve and the neural mechanisms that underlie these behaviors in a way that we would never have seen if we were only studying mammals in a laboratory. And so that's it. I want to thank my lab and my funding organizations. I also want to give a special shout out to Jessica Nowicki, she's a research scientist in my group, and she led a lot of this work, especially on the butterfly fish and the skinks that I showed you at the end. And I'd be happy to take any questions, and thank you for your attention.